Bismillahirrahmanirrahim. I am Dr. Jamil from the Physiology Department of Faisalabad Medical University, and this lecture is in continuation with endocrinology. And today we are going to start a new gland, that is the thyroid gland, that is present in the front of your trachea. This thyroid gland is very important metabolically, because if this thyroid gland is function normally, then you feel yourself active. If its function is decreased or there is a deficiency of its products, then you become lazy, you become obese. So let's start with the thyroid gland. The diagram is in front of you. It has got two lobes, right and left thyroid gland lobes, and it's connected centrally with an isthmus, and they are present in front of your trachea below the larynx. The thyroid gland secretes two hormones. One is the calcitonin, we will discuss it later on, and it also secretes the thyroxine, which is 93%. The T4 thyroxine is produced, and T3 is 7% of the triiodothyronine is produced. So both these things, both these products we are going to discuss today. There is the thyroxine and the triiodothyronine. The T3, the triiodothyronine, is an active form. It is 4 times more potent than the T4, but it remains in the blood for a shorter duration. This is very important. That although the T4 is more in quantity, but so far the activity is concerned, the T3 is 4 times more active as compared to the T4. But the T3, which is more active, more potent, and 4 times more potent than your T4, but its you can say its age or its limit to stay in the blood is far less as compared to the T4. Normally, the required iodine for its secretion is 1 milligram per week. Iodine in the form of iodide is required. Actually, iodide is an ionic form of the iodine, and when the two iodide molecules they combine together, they form an iodine molecule. The 1 milligram per week iodine in the form of iodide is required for the synthesis of the normal synthesis of the T3 and T4. That's why the table salt is iodized with the 1 part of sodium iodide to the every 100,000 parts of the sodium chloride. The table salt is sodium chloride, which is iodized with the mixing of sodium iodide, so that the iodide can be mixed with the sodium chloride, and this salt is required, although you can say the iodized table salt is required particularly in those areas with the deficiency of the iodine. Here is your next slide, the histological structure of the thyroid gland. This thyroid gland is made up of different follicles. Here you can see the word the follicle is written. That this is the central hollow place which is filled with the colloid, a gelatinous material, viscous material, which is known as the colloid. The purple color is shown over here, and this is the follicular cell. Actually, these are the cuboidal epithelial cells. Here is the follicular cells, and around these follicular cells, these are blood vessels. You can see the different side of the blood capillary. These are the blood capillaries you can see over here. So the formation of the T3 and T4 occurs in both this area, the follicular cells and the colloidal material over here, and the raw material, that is iodide, they are coming from the blood capillaries to this portion. So roughly these are the structure of the follicles: cuboidal epithelium, rounded, approximately rounded in shape, surrounding a viscous gelatinous material that is known as the colloid. Let's see what happens next. Now, this secretion of the thyroid hormone is basically dependent upon the thyrotropin releasing hormone. That is thyrotropin releasing hormone. It is coming from the hypothalamus. It binds with the receptor on the pituitary. Then pituitary releases TSH, thyroid stimulating hormone, which acts upon the thyroid gland, and thyroid gland begin to produce its products, the T3 and 4. Here are the things which are going to occur. The thyrotropin release hormone receptor on the pituitary. It will activate phospholipase 2 second messenger system inside the pituitary to produce phospholipase, calcium ions, and diacylglycerol, and so on. Now, the different steps. You can't understand all the steps through this written form. I will make you understand through an handmade diagram, but just for that, you can say making a notes or just to understand. The first step is in the production of the thyroxine or the T3, T4 is the trapping of the iodine through a sodium iodide symporter. Sodium iodide symporter. It was called the trapping of the iodide. I will see later on in details. Then the movement of the iodide into the lumen through a pendrin. Pendrin is a protein which acts as a channel through which all these iodides moves, and it is stimulated by an enzyme which is known as hydrogen peroxidase. Then production of thyroglobulin having tyrosine residues in the follicular cells is also stimulated by the hydrogen peroxidase. Then the oxidation of the iodides, there is two iodide molecules combined together to form iodine. Then the organification of the iodine, this is the incorporation of the attachment of iodine with the tyrosine molecule into a thyroglobulin. Now, this is the major steps for the formation of the thyroid hormone, the T3 and T4 in the follicular in the your viscous material in the colloid. After that, we will see the how it will the higher hormone is released into the blood. Let's see the next diagram. This is the handmade diagram through which you can understand all the things. Now you have to concentrate and you have to move along my cursor, along with my cursor. Now follow my cursor. The iodides, these are the iodides which are coming in the blood circulation. This is your capillaries. Here the pinkish color, the iodides, they are coming into the capillaries. Follow my cursor now. And the iodides which are coming in the capillaries, 20% of these iodides, they are moved into the follicular cell. Actually, this is the follicular cell. Here you can see the follicular cell. This is one cuboidal cell, and this is the center filled with the colloid material. This is the center which is filled with the colloid material. And this membrane, the greenish membrane, is known as the apical membrane. And this is the lateral membrane. This brownish is the lateral membrane, and this is the basal membrane. This is the basal membrane on this side. On the now this is the common structure. This is one, you can say the follicular cell is shown over the cuboidal cell. Now the iodides which are coming up into the capillaries, 20% of these are entered into the follicular cells, while the 80% moved out into the urine. 80% moved out into the urine. How these are entered into how these iodides they enter. Iodide is an ionic form of the iodine. It 20% they enters into the follicular cells through a sodium iodide symporter. Here is the pinkish circle showing the sodium iodide symporter. Actually, this is the iodide pump. This pumps the iodide inside, and at the same time it pumps two sodium also inside, which is not shown over here. The two sodium they also move over inside, and one iodide is also moved inside. So this iodide pump, or you can say sodium iodide symporter, is going to enable this follicular cells to accumulate 30% more than the iodide coming into the capillaries. That is going to accumulate more and more iodide inside this follicular cells. Next step. The first step is the entry or the trapping of the iodide into the follicular cells through iodide sodium symporter. Once the iodide is inside, then this iodide immediately moves onward, follow the direction, through a protein molecule which is a channel 1, this is known as the pendrin. Through this pendrin, this iodide moves inside this gelatinous material, the colloid. So once the iodide enters into the follicular cells, it moves directly through the pendrin molecule into the colloidal material which is present over here. Now Once it is present inside this colloidal material, it is immediately the oxidation of the iodide occurs. That is, two iodide molecules, they combine together to form an iodine. This is an oxidation process through which the two iodide molecules, the two iodide ions, they combine together to form an molecular iodine. Now let's back to this follicular cell. In the follicular cells, we have got different Golgi operators and the other ribosomal production. These ribosomes, they produce a protein which is known as thyroglobulin, TG, thyroglobulin. This thyroglobulin, which is produced by the ribosomes of this follicular cell, it also moves through this membrane and enters into the this colloidal material. So in the colloid, now we have got the iodine molecules. We have also got the thyroglobulin protein. Also we have got this thyroglobulin protein has got a very special thing, that it has got tyrosine residues. The tyrosine, here you can see the tyrosine residues. The tyrosine, these are the cuboidal structures. These are the tyrosine hexagonal structures. They are tyrosine molecules. So thyroglobulin, it produced by the ribosomes. It enters through the same channel, that is the pendrin molecule. It enters into the colloidal material. Now this colloid contains both the iodine molecule and a thyroglobulin molecule. Now these two things, the thyroglobulin is organic molecule, while iodine is a molecular structure. Now iodine is going to join this thyroglobulin protein, and this is known as the organification of iodine. That means the iodine has been joined to an organic structure. So this step is known as organification of the iodine. That means iodine has been joined to an organic structure. Now, this joining of iodine molecule to the thyroglobulin is very unique. Suppose this is a line of the thyroglobulin molecule. This is a line of thyroglobulin molecule having the tyrosine residues inside. 1, 2, 3, 4, 5, 6. Suppose these are the 6 tyrosine molecules present over here. Now, some of the tyrosine molecule will join with only one iodine molecule, one iodine join. This will known as the monoiodotyrosine. This is known as the monoiodotyrosine. Some of this tyrosine will be joined, some of the iodine will be joined by the, some of the tyrosine molecule will be joined by the two iodine molecules. These are known as the diiodothreonine. This will known as a diiodotyrosine. So this is your this molecule which is joined by the one molecule. This is known as the monoiodotyrosine, and this tyrosine molecule which is joined by the two iodine molecules. This is known as the diiodotyrosine. And then we have got the same repetition. It may be the one molecule, it may be the two molecule, and it may be the two molecule, or it may be the two molecule. After that, then there will be pairing of these molecules will occur. For example, the molecule having one iodine molecule, it is going to pair with a molecule having the two iodines. So that means monoiodotyrosine is going to pair with the diiodotyrosine. So if these two combine together, if these two pair is going to form, this is known as the T3. And if the pair is formed between the tyrosine having two iodine molecules and another tyrosine having further two iodine molecules, that means two tyrosine molecules having two iodine molecules over each. So this, if the pairing of such a molecule occurs, it will be known as the T4. Here T is missing here, this will be known as the T4. So again, repeated, trapping of the iodides through sodium iodine symporter, then movement of this iodide through pendrin into the colloidal material where the oxidation of iodine is going to occur. The two iodine molecules, they are going to join, two iodide molecules going to join to form an iodine. Then this iodine is going to join with the thyroglobulin, which is produced by the ribosome within the cell. Now this thyroglobulin molecule, they have got the multiple tyrosine residues, amino acid tyrosine residues, they are present here. Now this union of, or you can say this organification is haphazard. Some of the tyrosine molecule will receive only one iodine, some will receive the two iodine molecules, some, and after that, and then there will be pairing of these tyrosine molecules will occur. If the pair is going to occur between one molecule, tyrosine having one iodine and the tyrosine having two molecules of iodine, then it will form the hormone which is known as T3. And if the pairing occurs between the tyrosine molecule having two iodines and another tyrosine molecule again having two iodine, then it will make the hormone T4. Now, all these things, that is all these things, that MIT, monoiodotyrosine, diiodotyrosine, triiodotyrosine, and T4, all these things, they will come back into the cell, into the follicular cell. They will come back into the cell. In the cell, suppose they are coming into this cell. Now, the lysosomal proteolytic enzyme will act upon these things, because all this molecule having T3, T4, DIT, MIT is back to the follicular cell. In this follicular cell, lysosomal proteolytic enzyme is going to act over each other, and it will release MIT, DIT, T3, T4. And, or you can say in other words, these lysosomal proteolytic enzyme will separate MIT, DIT, T3 and T4 from each other. Now this T3 and T4, they will leak into the circulation, back into circulation. Now T3 is formed, the arrow is going down, T3 is formed in very less amount. While T4 is formed in larger amount, the arrow is going up. Now what happens to MIT and DIT? This monoiodotyrosine and diiodotyrosine, they are acted upon by the thyroid deiodinases. This is the name of an enzyme. It will remove the iodine. It will remove the iodine from mono, MIT and DIT, and this iodine will be recircled and go back into the colloidal material. While this proteins will be also moved, the protein which is remaining here, that will also move back into the circulation. Now, name of the different enzymes which are acting upon this area. This iodine, oxidation of iodine, or you can say first of all the movement of iodide through pendin into this area, and then oxidation of this enzyme, oxidation of this iodide molecule, and organification of this molecule, and then the pairing of these molecules. All these things are accelerated by an enzyme which is known as hydrogen peroxidases. Hydrogen peroxidases is very important enzyme which is going to accelerate the reaction of the oxidation, organification, and at the end your pairing. This pairing of this area is, all these actions are accelerated by an enzyme which is known as the hydrogen peroxidases. And here we have got the lysosomal proteolytic enzymes which are going to cleave the T3 from T4, MIT from DIT. Then another hormone which is known as the thyroid deiodinases, they are going to remove the iodine from this MIT and DIT, and it will be recircled from here. Now, once the T3 and T4, they are leaked into the circulation, they have to reach into the tissues. Now you must know that T3 is the active form. T3 is active form and T4 is not an active form. This T4 is not an active form. T4 has to convert into T3 before it becomes active. Although the T4 is larger in quantity, T3 is less in quantity, but the active form is T3. So what happens? Another enzyme which is known as the 5' mono deiodinase enzyme. It will remove 5 iodine from the 5' location. This is the 5' location, and this T4 is converted into the T3. Sometimes what happens, another enzyme which is not 5 prime, it is just 5 monodeiodinase enzyme, it will remove the iodine from this position. And if the enzyme is removed from this position, this is known as the reverse T3. This is known as the reverse G3, which is an inactive form. We will see in detail this reaction. The conversion of T4 into T3, we will see. Let's see another diagram of this area. Here you can see the 5 prime deiodinase enzyme convert T4 into T3. Here this is your T4. This is the formula of the T4. This is the 5 prime location. This is the 3 prime. This is a simple 5. This is a simple 3. This is the formula of the T4. Now the enzyme which is known as the 5 prime deiodinase is going to act upon this area, and it will remove this. Follow my cursor. It will remove the iodine from the 5 prime position, and the next molecule will be this, that you will see that at the 5 prime location there is no iodine. So this is the structure of the T3. This is your T3, which is active form, while another enzyme which is just known as the 5 monodeoxidase 5 deiodinases 5 deiodinases, it will remove the oxygen not from the 5 prime but from the simple 5. So next thing is formed like this, and this is the formula of the reverse T3, which is an inactive form. We will see later on the other details. At this stage, you must know that how the T4 is converted into the T3, which is an active form, and how it is converted into reverse T3, which is an inactive form, reverse T3, and this is by the action of the enzyme. T3 is formed by the 5 prime deiodinase, while reverse T3 is formed simply 5 deiodinase, which convert the T4 into the reverse T3. And this is a diagram of your book. Let's repeat the things from your book. That here is the sodium iodide symporter, yeah, you can say iodide sodium symporter, which moves 2 sodium and 1 iodide inside. Yeah, you can say this is known as the trapping of the iodine. Just wait a minute for it. Here's your sodium iodide symporter. Once the iodine is inside, it will move through pendrin. Actually, when the pendrin is working, it moves 1 chloride ion into the cell. This iodine, it will move inside, and there will be oxidation of the iodide. It will form an iodine molecule. Then this iodine is going to combine with the thyroglobulin, which is known as the organification. And after the organification, when this organification is going to occur, there is the formation of MIT, DIT, T3. Here, the reverse T3 is actually not going to form over here. Some books say the reverse T3 also can be formed over here, but usually it is formed in the peripheral tissue, then T4. Then what happens? The apical membrane, it will form a, you can say this is a pinocytosis process. It will take a bite of this thyroglobulin. This thyroglobulin will contain all these things, and then when it enters into over here, the proteolytic enzyme is going to act upon it. They will release T3, T4, which will move into the blood supply, while that MI and DIT, they will be acted upon by the deiodination, and the tyrosine molecule residues. Both of them, they are recircled, and again the things move in. Although this diagram is quite deficient, the handmade diagram by me is much more elaborated, and you can understand the things very easily. Now, to write down the things, you must know the steps how you can write down things. Number 1, thyroglobulin synthesis by the ribosomes, then uptake and concentration of iodide by the sodium iodide symporters, then oxidation of iodide to iodine molecule, then iodination, or actually this is the organification of the thyroglobulin, and then there is the formation of MIT, DIT, and at the same time T3, T4, and ultimately there is a secretion of T3 and T4. So dear students, this was all about the formation or the synthesis of the T4 and the T3, and later on we will see in the next coming lecture that what are the functions of these T3 and T4 and how they are act upon your body. Thank you very much.